All right, guys. So at this point, you should have completed the Discovering DNA Structure activity. So we are going to talk about this and go through and learn about DNA structure as we go. Um, so starting off, you put together a nucleotide and colored it. And if you're at home, you may or may not have done this. But hopefully you can look at the structures and see how they work together. But three common parts of a nucleotide. You go ahead and check your work as we're going through it. But the three common parts, one nucleotide looks like this. So it has a deoxyribose sugar, a phosphate, and a nitrogen base. And the part of the nucleotide that changes to create the four different nucleotides. So here's one nucleotide here. We have another one here, and then another one here. And the difference in all of these nucleotides is this part. You can see how the colors change, and we have four different nucleotides. So the nitrogen base is what changes, and the names of the nitrogen bases are in DNA are adenine, thymines, cytosine, and guanine. And on one strand of DNA, when they talk about a strand, they're talking about one of the sides of this ladder. So when they talk about a strand of DNA, they're talking about this, and they want to know where one nucleotide connects to another nucleotide, and that would be right here between the phosphate and the sugar. And the bond that holds them together is a really strong bond, and if you remember way back in August, we learned about ionic bonds, hydrogen bonds, polar covalent bonds, and nonpolar covalent bonds, or just covalent bonds, and the strongest of those was the covalent bond, where the electrons were shared equally. And that is what we see between the phosphate and the sugar. It's a very strong bond that helps hold these molecules together. So when you're looking at this DNA molecule, if there's strong bonds between those phosphates and sugars, it's not going to come apart real easily. It's going to hold those bases together. And where do the two chains of nucleotides bond together? So now they're talking about this chain and this chain. Where do they connect? Well, they connect here between this base and this base. So the nucleotides come together between the nitrogen bases. And the bonds that hold them together are weak bonds. And the weakest bond we talked about was a hydrogen bond. And those hydrogen bonds are weak, so that tells us that these two strands, this strand and that strand, could very easily separate from each other, whereas the bonds between the phosphates and the sugars are going to stay together and they're not going to come apart. So that allows us to talk about these bases a little bit. The order of the bases is very, very important. That is what determines which proteins are made. So if the order of these bases, A's, T's, G's, and C's, if they're important and it determines the protein, it would make sense that we don't want the bonds between these nucleotides, between the phosphates and sugars, to break, because we want to keep them in a specific order so that we can make a specific protein. Now looking at this picture, it said, the question is, what is the pairing arrangement for nitrogen bases? Well, every time we see a G, it is attached to a C. So guanine always pairs with cytosine. And wherever we see an A, we see a T. So adenine always pairs with thymine. All right. Then they ask, are the molecules of adenine always going to equal the number of molecules of thymine? And so let's look at our diagram. Let's look at our picture right here and look at that. We have 1, 2 A's and 1, 2 T's. Will that always be the case? Yes. Why? Because wherever there's an adenine, a thymine is going to attach. Adenine cannot, if you look at the pictures, an adenine can't attach to a guanine or a cytosine. It can only attach to a thymine, and it's more stable when they are together. So an adenine will always attach to a thymine, so yes, they will always equal each other. And then it says, well, will guanine equal cytosine? So if we look here, we have one cytosine, two cytosines, one guanine, two guanines. So will they always match, or will they always be equal? Yes, for the same reason. The guanine can only attach to a cytosine, and cytosine can only attach to a guanine. Now, scientists abbreviate the letters, abbreviate these words with letters, which we've been using, but A always binds to T and G always binds to C. And when we see those letters A, T, G, and C, we know that we're talking about DNA. And if I had this, if this was one side of a DNA strand, ooh, not that. That's one side. What would the other side look like? Pause the video, see if you can fill it in. We would get what is called the complementary strand, following what we call the base pairing rules, A to T, G to C. So the blue is the complementary strand to the A, creating our double-stranded DNA molecule. Now remember, these are just the bases. They are attached to a sugar and a phosphate. Every one of these to make a nucleotide, but the bases, the order of the bases is what we look at when we're trying to figure out what it's coding for. All right, the shape of DNA is called a double helix. And that name and the structure of DNA was discovered and credited to Watson and Crick. The double helix means that there's two long chains of nucleotides arranged in a spiral or twisted ladder-like shape. Sometimes it's also called a spiral staircase, but double helix is the actual name of the shape of DNA. The sides of the ladder are made up of alternating phosphates and sugars. And the steps of the ladders are made up of the nitrogenous bases. So this right here, the sides where the phosphate and sugar is, it is called a phosphate sugar backbone. I don't know, my handwriting is horrible, I'm sorry. That's called the phosphate sugar backbone. And we see that in all nucleic acids, whether it's DNA or RNA. The phosphate sugar backbone is the main component of it, where the bases come off of that phosphate sugar backbone. And the sugar in DNA is deoxyribose. And DNA stands for deoxyribonucleic acid. All right, so based on what we've learned from this activity, it's time for you to complete activity pre-AP lesson 4.1. You're going to read, look at the data, complete the graph. It will be a bar graph. And then complete the part 2 at the bottom. Pause the video when you've completed working on that. You can unpause the video and we'll talk about a few things that you need to make sure you're aware of. So now that you've done this, some observations that you might have made. You might have noticed that the amount of A and T is greater than G and C in all of the different organisms. You might have noticed that the sea urchin tends to be slightly different than all the rest. It's kind of the outlier of the group. You may have also noticed that the amount of A is pretty similar to the amount of T. The amount of G is pretty similar to the amount of C. And that all of these, if you add them up, it equals approximately 100. Now, not exactly 100 because of rounding, but those observations you may have made, there may be others that you made. The more observations you made, the more you analyze the data, the better off you are. Good job. Let's look at how we would have set up the graph, and you grade yours as we look at it. Your title, it should be something about the percent of nitrogen bases in DNA, and you might have put it in DNA of different organisms. Your y-axis should have had the percent. And on your x-axis, I would have set it up with the organisms at the bottom. There are other ways to do it. To me, this is the most efficient, where you're going to divide it out by organism. So you have human, chicken, grasshopper, and urchin. And then in each of these, I would have put a bar for each of the bases. My scale, whatever you decided your scale would be, 5, 15. Now, I know this looks kind of off, but it's really counting by fives by squares. So 5, 10, 15, 20, 25, 30, 35, counting by squares as we go. And we would keep going to make sure we filled it in. So you would choose a scale that would work for the numbers that you have. And then I would have created a legend. So A, I would have done in red, maybe T in yellow, maybe G in green, and C in orange. And then for each organism, you would draw a bar that would go up to the amount indicated by the data. So for each organism, you would have 4 bars, and what you would notice is that the A's and the T's would be basically the same, and the G's and the C's would be basically the same. Now this is kind of hard to draw on the board. That should be orange. So use this as a guide and grade yourself. If you didn't do it, pause the video right now and attempt to do this graph. It's good practice for you. The more graphs you draw, the better you get at it. All right. Well, then if you look at part 2, it asks you to make some claims and inferences about part 1, and we did that when we looked at the data, and now we're going to use what we saw to fill this in. And remember, what we figured out is that A equals T and G equals C. So for T, since A equals 27.3, T would equal 27.3. And since G equals 22.7, C would equal 22.7. And that does equal 100. Now in some of these, we're going to use the same idea, 32.5 and 17.7. And it's a little bit over 100, and that is okay. I think it's like 100.2. So you could adjust the numbers, or you can leave it right about there, just because we don't know the exact data. All we know is that the amount of A is supposed to equal the amount of T. 24.1 and 26.0 for this one. So that's what we're looking at. If you adjusted it up or down a little bit to make it equal 100, that is absolutely okay. As long as we can see that you know A and T are supposed to be pretty similar number, and G and C are supposed to be pretty similar number. So now, let's say we have a dog, and we looked at its DNA, and this is the information you're given, that the amount of adenine is 35%. Can you fill in the rest? So adenine is 35%, so we know A and T have to equal, so thymine would have to be 35. Well, 35 plus 35 is 70. Total percentage possible is 100. So 100 minus 70, we have 30% left to use for G and C. Well, we know they're supposed to be equal, so it would be 15 and 15. Those are just some examples of how we might apply these numbers. And there's a few more terms we need to talk about while we're looking at, while we're talking about DNA structure. So let's go through and talk about some of those. But first, let's review some things we know about DNA before I give you the last few terms. One, it's double-stranded. sugar is deoxyribose. It's made of nucleotides. And nucleotides have 3 parts: a sugar, a phosphate, and a base. A always bonds to T, G always bonds to C. The amount of A equals the amount of T. The amount of G equals the amount of C. It has a sugar phosphate backbone. And its shape is called a double helix. Now, let's look at the structure. You might have also noticed when you were putting together your structures that the strands go in different directions. So this strand, the phosphates point that way, whereas this strand, the phosphates point that way. That is very, very important in the way DNA is built. And then you notice these numbers, 3' and 5'. Well, it has to do with how we count the carbons. And if you can look here, when we look at the carbons in the sugar, there's carbon 1, 2, 3, 4, and 5. The phosphate is attached to the 5', fifth carbon, so we call it the 5' end. The end with no phosphate, the end that attaches to another nucleotide, is the 3rd carbon. And so here there is no phosphate, we call it the 3' end. Now, a way to remember it, the end with the phosphate has more, so we use the number that is more. 5 is more than 3. So the 5' end will always be the end where the phosphate is. The 3' end will always be the end where there is no phosphate. All right, so those are just some basic things about the structure of DNA. Let's talk about a couple of terms. Some of them we've mentioned before. The phosphate sugar backbone, that is covalently bonded deoxyribose to the phosphate group. And it makes for a very strong structure to where it's not, the length of the structure is not breaking. Complementary strands follow the base pairing rule where we have A, T, C, G, A, T, and C. So the complementary strand would be what complements it, what goes well with it, what can attach to it. So those are our complementary strands. The base pairing rule says that A binds to T and G binds to C. Then we have Chargaff's rule. And we haven't mentioned that before, but we've talked about it already. Chargaff is a chemist, and he did tests on DNA when they were trying to determine the structure. And those tests were used to determine, or his test determined that the amount of A equals the amount of T, and the amount of G equals the amount of C. Anti-parallel says that DNA strands go in opposite directions, and the end with the phosphate will be the 5' end, and the end without the phosphate is the 3' end. Now this becomes very important as we do DNA replication and we build DNA and RNA. But anti-parallel says that those strands go in opposite directions from each other. All right, so now hopefully you printed your CFA out. If not, you can open it up and answer on a piece of notebook paper, but you're going to go through the CFA and answer those questions 1 through 11. Pause the video while you answer those. When you're done with that video, you can hit play and check your answers and see how you did. So the first question says, which statement below best summarizes the role of DNA molecule in cells? And the correct answer is C. It does provide the instruction for making proteins. What regulates the processes that provide the cell with energy? That's the mitochondria. What guides the cell division? That is the centriole. And what protects the cells from infection? That would be the cell membrane. Which statement is true regarding DNA?
All students may now exit campus. Remember to stay 6 feet apart and wear your mask correctly. Walk directly to your designated area.
It does contain deoxyribose sugar. A is the correct answer there. B, C, and D are all descriptions of a nucleic acid, but of RNA. Number 3, all but one of the following is a component of nucleic acids. Nucleic acids do not contain, which of the following? It does not contain an amino acid. The other 3 are contained in it. Ribose is the sugar in RNA, but RNA is a nucleic acid. The sequence of blank in a DNA molecule determines the protein that will be produced. The correct answer is nucleotides. And the nucleotides, the order of those determines the order of the bases, which determines the order of the amino acids in our proteins, which determines the traits that are produced. A segment of DNA that codes for the ability to make one type of protein molecule is known as a gene. That's going back to what we just talked about in the previous unit. A gene is a segment of DNA that has the code for a specific protein. The function of DNA and where it is found in a eukaryotic cell. Well, we know DNA in a eukaryotic cell is in the nucleus, and it does regulate all cell activities. It regulates those activities by coding for the protein, and those proteins can be enzymes. Those enzymes regulate what happens in and out of the cell. Number 7 says a scientist is purifying DNA from a bacterium, and we want to know which of these molecules could have been DNA. Well, we know DNA has a sugar, it has phosphates, and it has bases. And guanine is one of those bases. So molecule 3 has all 3 of those. So molecule 3 is the DNA. Number 8 says, what is deoxyribose? Well, OSE tells us that it is a sugar, so B is the correct answer. Deoxy tells us that it's in DNA. If it was just ribose, it would be the sugar in RNA. Then it says in DNA, molecular ratio of adenine to thymine is 1 to 1. If a strand of DNA there are a million A molecules, then how many T molecules? Well, with a ratio of 1 to 1, that means for every A we have to have a T. So if we have 1 million A, we have to have 1 million T molecules. Hopefully you're doing well so far. Number 10 said in the nucleotide pairing, adenine pairs with thymine and guanine pairs with cytosine. The table shows the percentage of adenine and guanine, cytosine, and thymine in a rat, and we want to know which of these is correct. Well, these choices just set up a ratio, a division, and we're saying, so if we divide C by A, we would get approximately 3. Or B, if we divide G by T, we get approximately 2. C says if we divide T by A, we would get approximately 1, and D, if we divided G by C, we get approximately 4. So if we look at the numbers and do the division, we would see that this is the only one that works. C is the only one that works. But we also know that T is supposed to equal A. So if we were to divide something that is equal to itself, or something that was equal to something else, we would get a ratio or an answer of 1. And number 11 says both DNA and RNA nucleotides contain 3 basic components. The nucleotides contain all of the components listed except amino acids. Oh, not him. Amino acids are the monomers of proteins, not of nucleic acids. So I hope you did well. Put how many you missed. Take a picture of it and email it to me with the pictures of your discovering DNA activity and your investigating the molecules of DNA. But send pictures of all of that to me so that I know you're keeping up with the work and doing the lesson. And that is it for structure of DNA. Hopefully you're okay. If you have any questions, just send me an email and I will gladly respond.